Okay, welcome to Unit 2. Give it up for Unit 2. I'm excited to get out of Unit 1. Chemistry of life is somewhat interesting, but we start getting into the good stuff now. This unit is cell structure and function. The first bit, we're mainly just going to be talking about the cell structure, so organelles, all that kind of stuff. Spend a lot of time at the cell membrane. And then we'll talk a little bit about compartmentalization towards the end. Not all in this video, just kind of look to the unit ahead. So what we're going to be doing in this video is taking a bit of a tour of the cell, starting off with types of cells, and then diving into their organelles. So, fundamental unit of life is the cell. It's the simplest collection of matter that we consider to be alive. Whether or not something is alive is a very tough question. In reality, creating any definition that perfectly suits what you want is impossible. We'll do an assignment in class where I ask you to try and write a definition for a chair, and we'll see how that goes. An important thing to know with this is that it's all based on the idea that all of our cells are coming from pre-existing cells. So everything is related, all life is related, and even though cells can be very different in their look and function, there are a lot of things that are common between them. So let's start with what is common between all the cells. So first, the main things are plasma membrane, having a cell membrane. Kind of need it to have a cell. You need to have an inside and an outside. You need to have cytoplasm. You need to have something filling up the inside. You need to have some sort of genetic material. Most things use DNA. Keyword is most. And then ribosomes, that structure that actually makes your proteins. So everything has those. The plasma membrane specifically is a phospholipid bilayer. You should remember phospholipids from last unit. And it's full of proteins that separates the cell from the surrounding environment. You'll sometimes see it called the fluid mosaic model, with the embedded proteins making up that mosaic. And the purpose of the cell membrane is to control what goes in and out of the cell. It's what we call semi-permeable, meaning it lets some things in, while other things need extra help to cross the barrier if they're even able to at all. Cytoplasm is all of the goo inside the plasma membrane. It's the cytosol and all the particles inside of it, mostly water, but full of proteins and other things. Everything has chromosomes with some genetic material. As I said before, it's almost always DNA, but it is sometimes RNA. In very small exceptions. There's some people that believe that RNA is the original genetic material and that DNA came second, rather than the other way around. But your genetic info is the coding for your functioning, essentially for all your proteins. The ribosomes are your protein factories. They are made up of ribosomal RNA, a special kind of RNA, as well as some protein, made up of two parts, but their main focus is to just build proteins using the instructions coded in the DNA. Generally, the rule of thumb is the more proteins you have to make, the more ribosomes you're going to have. So a pancreas cell, for example, it helps make digestive enzymes, and it has a few million ribosomes in a single cell, which is part of why it is such an awful organ to get cancer in. Because it makes so many proteins, it spreads very quickly. Whereas other cells, like your skin cells, are not really making many proteins. And so cancer doesn't spread as easily in skin cells. Okay. Everything on Earth we will consider either prokaryotic or eukaryotic, and you do need to know the difference. So prokaryotic cells are your single cell organisms. I call them prokaryotes, and the main thing that sets them apart is they do not have a true nucleus. They still have genetic material. It's just not in its own compartment in the nucleus. Eukaryotes are single cell and multi-cell organisms. So just because something is single cell does not mean it's a prokaryote. It could be a eukaryote. And they're generally a little more complex. They have a nucleus and they have other membrane-bound organelles, so they have just more organelles than any prokaryote. And they're generally more complex, but they can be very simple. Don't automatically think they are complex life. So what are the differences? Again, prokaryotic cells are simpler, smaller, just less going on. They have a cell membrane, they have cytoplasm, they have ribosomes. Instead of a nucleus, they have a nucleoid, which is just the region where the DNA is. It's not surrounded by a membrane like eukaryotic nuclei are. And importantly, and we'll talk about it a lot more later, the chromosomes are circular instead of linear, and that'll come up a lot in Unit 6. They also have a cell wall. They need to generally keep their structure. Single cell things, if they don't have a cell wall, struggle to keep any sort of structure. Think of an amoeba and all of the different shapes it can take on. So a lot of prokaryotes will have a cell wall, but it is not made of the same thing. It's not made of cellulose, it's made of peptidoglycan. It's still a carbohydrate, just a different one. A lot of them have a capsule on them. It's just kind of like a jelly coating. And then flagella help them to move around. Eukaryotic cells are where we're going to be spending most of our time. They're more interesting, to be honest. They are generally larger and generally more complex, but not always. They also have a cell membrane, cytoplasm, and ribosomes. However, their DNA is enclosed in a nucleus in a second membrane. So the nucleus itself has a membrane that keeps things from inside the cell out of the nucleus. And our chromosomes are linear instead of circular. Additionally, eukaryotic cells are going to have a ton more of what we call organelles. They're essentially a bunch of specialized compartments. And the idea of compartments is going to be a big theme for the next bit in this class. Compartments let you specialize. And specialization is how you get a lot of complexity and multicellular life really emerges. But it starts with these organelles. So they will be membrane-bound, surrounded by their own membranes. Think your mitochondria, think about chloroplasts, think endoplasmic reticulum, and they all have very specific functions. We will be talking about these in the next section. So I'm going to move on for now. All of this builds into the idea of we have 3 domains of life. These change all the time. When I was growing up and in high school, we didn't have 3 domains of life. We had the 5 kingdoms of life. So this science of taxonomy is really a constantly changing process. There's no good way to actually group everything in our thing. Life just doesn't fit into categories. Where we will try and we can put things into these groups, but ultimately it's messy. And trying to put it into these clean, neat groups just doesn't really work the way we want it to. But the current understanding is that the grand ancestor of all life gave rise to kind of 3 separate lines. Bacteria, which are prokaryotic cells, archaea, which are also prokaryotic cells, and then eukarya. Eukarya are closer related to archaea, they believe at least. And then from eukarya, we get the diversity of all of the life that we see on Earth, from fungi, plants, animals, and protists, kind of the single-cell eukaryotic cells. In 10 years, this will likely be different, so I wouldn't worry too much about it. What you do need to know are some differences between plant and animal cells. So, both eukaryotes, but there are some structural differences. Firstly, plant cells have a cell wall made up of cellulose, gives them that crunch, gives them their structure. It's kind of their skeleton. They also have a large central vacuole. A vacuole is kind of just a container for stuff. In this case, it's water that helps it kind of keep its shape and turgidity. And they'll also have chloroplasts, which do photosynthesis, all of which are absent in animal cells. Animal cells have centrioles, which are kind of a cytoskeleton structure. We'll talk about them later. As well as lysosomes, which help to do some cellular digestion, both of which are absent in most most plant cells, but not all. So, general overview of a plant cell. We are going to do an activity regarding these organelles within a cell. I'm going to play a cell dating game. It's going to be a lot of fun. And we will dive into the rest of these organelles in the next little bit. So, interactive note questions. Remember, these may come up as quiz questions. I may pull them for practice problems. I do pull them from practice problems a lot. Okay, I'm going to do a quick time check. So it's going to freak out on you for a second. How long have I been recording? 11 minutes? We're good. Okay, going to continue into the endomembrane system. So, the endomembrane system is